Hi, this is Liz Harvey. I am an assistant professor at the University of New Hampshire, clearly quarantined in my house. Because we all got kicked out of our labs, I had to bring my phytoplankton algal culture collection home with me. And I thought, because I'm really bored, I would make some kind of video about me transferring my algae. I've heard a lot from other people when I talk with them, and they tell me that they think culturing is really hard. It's not that hard. Clearly, you will be able to watch me do it from my house. Back in the day, people used to have victory gardens, I think during the World Wars, and so this is my homage to the victory garden, but in this case, it's all of my research phytoplankton. So I have no idea what I'm going to say. It's probably going to be super awkward, but we'll just go with it. So the first thing that I'm going to do is transfer all my algal cultures. So for those people who don't know, these are phytoplankton, which are single cell plants essentially that live in the ocean. They conduct photosynthesis like trees and grass and other organisms that you think about, as plants living on land. They just live in the ocean. And they produce about half of the oxygen that you breathe. So they're really important in that respect. If you enjoy breathing, you probably should appreciate phytoplankton. Also, they're the base of the marine food web. And they're really important in terms of how carbon and nutrients and other chemicals are cycled throughout our globe, so important in global climate and things like that. In my lab in particular, I am interested in how phytoplankton die, which I know seems super morbid, but it's actually really interesting. We know a lot about how phytoplankton live. So they need light and nutrients just like land plants. So if you could go out in the ocean and spray a bunch of Miracle-Gro, you probably would get some phytoplankton to grow. That's a simplification of how it works, but it's sort of the idea. But they can also die, and they die in lots of different interesting ways. So they can get eaten by other single-cell grazers called microzooplankton. They can get viruses, just like you and I can get a virus. Clearly, we're living that reality now. Phytoplankton can also get viruses. Most of the time, they die from viral infection. Phytoplankton can also have really interesting interactions with bacteria. And they can exchange chemical compounds, and some of those chemical compounds can cause mortality. Phytoplankton can also get a parasite, which is kind of cool, and it replicates inside the phytoplankton and explodes out, killing the phytoplankton. All very sad. However, to do all of this work, we need to have some cultures that are sort of our library collection that we use for laboratory studies, and that's what's here now. So the way that I keep them going is every 2 weeks, I exchange the library. So I put some new phytoplankton media. This is seawater that I have previously collected and put some nutrients in to help them grow. I put some new fresh seawater in some fresh tubes, and then I'll transfer a little bit of the algal cells, and I have them growing in the window of my foyer. They seem to be doing okay there. We'll see after this transfer. And then also here, I have some of the grazers. So I also keep in my collection some of the grazers, and so we're gonna I feed those as well. They get grown in filtered seawater, and then I feed them. So it's all pretty standard, and because we had to leave everything in our lab. But I was able to bring home some equipment. So this is an automatic pipetter. It makes pipetting water very easy. Normally, you should be doing this in a hood that protects the cultures from getting contaminated. Mostly, people are concerned with bacterial contamination, but in this case, we're going to make do with what we have because clearly nothing is going to go as normal in these days. So right now I'm putting in the seawater into the vials, and then in a minute I will transfer my phytoplankton. And you can, people can do this in their home. I don't know. Back in the day, in like the Victorian era, people used to have parties where they would look at phytoplankton under microscopes and make detailed drawings of beautiful diatoms and things like that. But I guess we got away from doing that for some reason. I don't know. I'd like to be invited to one of those parties. So the way that phytoplankton work, there's a couple different kinds of phytoplankton. The three major groups are called diatoms, coccolithophores, and dinoflagellates. And I have all of them in my culture collection, different species. Diatoms live essentially in a glass house. They have a glass frustule or shell that surrounds their cell. Coccolithophores live in a calcium house. They have a shell or are surrounded by calcium carbonate liths. And then dinoflagellates have flagella and they can swim. So that's sort of their claim to fame. And if you can see these cultures, I'll hold up a couple in a second, you'll see that some of them are different colors. That's due to the pigments that they have inside their cell. So for example, this particular species is brown and this one is green. You can see those pretty easily. The way that we isolate phytoplankton is by going to a water sample and we essentially catch a single cell and we put it in some growth media and hope that it grows. And then it generally reproduces asexually and we're able to keep it in a culture. So all of these cultures were started by a single cell, and then the cultures just divide pretty much once a day if you're lucky. Probably now they're not in the most ideal conditions, so I'm not sure they're dividing exactly once a day. But right now I'm just taking, so I put 25 milliliters of my seawater growth media in there, and I'm putting 1 milliliter of my culture, and hopefully that will get them growing again and they'll be super happy, and I will not lose any time or cultures with coronavirus. I brought home enough stuff to comfortably culture for probably 8 months. If we're gonna go longer than that, then we're gonna have to get a little more creative. Hopefully that's not the case, but I was a little manic when I packed all my stuff, so I took as much stuff as I could get. And my husband and my son have been very kind in sharing the house with all my science equipment and everything too. So that's pretty much it. I'm gonna just keep going here and culturing everything. If you have questions, you can find me on Twitter or email me at elizabeth.harvey@unh.edu. I'm happy to chat with anyone who is interested in marine microbes. And hopefully this wasn't too weird or awkward. I'll probably never watch it back in full because I hate the sound of my own voice. Hope everyone stays safe and hope to see everyone soon. Bye!